Assalamu alaikum. I would like to welcome you all to the first virtual launch meeting for Sandoz Transplantation Portfolio in Egypt. الحقيقة we've decided to go with the virtual launch rather than to wait until we meet face-to-face soon, inshallah, not to waste any time to bring this high-quality medication to the Egyptian patient and to the Egyptian doctors. It's really such a great honor to have you all here today. Thank you very much for your time. And I'm really privileged also to have such a distinguished panel with us. So please let me introduce the panel. We have such very good speakers and panel with us, Professor Mahmoud El Meteini, and he's a professor of hepatobiliary liver transplant surgery. And he's, of course, the president of Ain Shams University. Professor Effat, he's the president of the Middle East Society of Organ Transplantation, and he's an elected council member of the International Living Donor and Living Transplantation Study Group. And from overseas today, we have Professor Rita Allowy. She is a research professor of medicine and director of transplant clinical research in the University of Cincinnati. She in the College of Medicine, Department of Internal Medicine and Nephrology Department and Kidney Care Program. And we have also the pleasure to have Dr. Amit Kumar Rai. He's the Global Medical Manager for Transplantation Sandoz in Europe from overseas as well. And we have also Dr. Peter Effat, the medical head for Sandoz Egypt. All this panel today, our intent is to launch the Sandoz portfolio, to let you know about the portfolio. And we're really looking forward to such a great night with the panel and with your interaction. Over to you, Professor Mahmoud.
Good evening, everybody, and thank you very much for inviting me as a panelist in this virtual meeting with Sandoz. And I'm really glad to be with this with my dear friend Professor Effat Kamel and from our friends from overseas, very distinguished panel, Dr. Rita Allowy and Dr. Amit, and from Egypt. My dear old friend, Mr. Sameh Al-Bagouri, and thank you very much, Sameh, for the kind introduction, and Dr. Peter, Mohammed, and all the Sandoz team. I have been asked with my friend, Professor Effat Kamel, to set the stage for the distinguished two speakers to present to us at the launching today of a very important generic for the tacrolimus that we have been using in Egypt over the last nearly 20 years now, since we began for the very first time our liver transplant programs in Cairo, Egypt, that spread all over Egypt later on. Actually, I'm not sure if we about the topic that we've chosen, the new perspective of immunosuppression after 19 years. Tacrolimus and the CNI at large has been used heavily in liver and kidney transplant, the only two solid organs that are being transplanted in Egypt, because we are only doing living donors. We don't have yet a cadaveric program, very unfortunate, but this is a fact. So we're using, we're making a kidney transplant for the last more than 30 years, and liver transplant, as I said, 19 years ago, and we used all kinds of CNIs. Of course, in the liver world, we are ebbing most towards the tacrolimus rather than the cyclosporine. Liver transplantation, as you all know, is the only credible long-term treatment for end-stage liver disease, whatever the cause is. The main indication for liver transplantation worldwide has been end-stage liver disease, mainly due to viral hepatitis, and 50% of the transplant all over the world, in the United States, in Egypt, 50% in the United States, but in Egypt it's more than 90%, were due to end-stage liver disease related to HCV. Of course, the landscape has been changed over the past few years and expected to change more and more in the coming years after the appearance of the DAs over the last few years. Other causes of end-stage liver disease evidently is metabolic diseases and hepatocellular carcinoma, as well as alcoholic liver cirrhosis. Although this cause, as a cause of end-stage liver disease, it's not really widespread in our part of the world, like in the western part of the world. And the last years, we were very fortunate, the last, I would say 20 years or more, that the liver transplant society have been enjoying a 1-year survival rate, as you can see, around at least 85 to 90% with an operative mortality that is 3 months mortality only in the terms of 5 to 10%. And if we come to Egypt, the largest percentage of liver transplantation in the Arab countries, the Middle East, I would say, has been performed in Egypt, with a majority in Egypt being done, or 100%, not the majority, LDLT, and no cadaveric liver transplants at all. The majority of the cases in Egypt have been done in 2 centers, who are the 2 centers that really started back in 2001, Wadi Nil Hospital and Dar al-Fuad Hospital. Dar al-Fuad, where Professor Effat is working with his team, and our friend there, and Wadi Nil, where I started with my team, and the late Professor Ala Faiz. Said that, I would say that in our other Arab countries, like, for example, in Saudi Arabia, there are a good percentage, or at least increasing numbers in the cadaveric programs, and they are ahead of us in this. How many liver transplants have been done in Egypt from 2001 to date? We are approaching the 4,000 LDLT over the last 19 years. In how many centers? We started by only 2 centers, and now we are bypassing the 14 centers. I'm not sure this is well geographically distributed. It's a wrong decision, right decision. We can discuss this later on, but at least this is the fact now. As you all know, the best immunosuppressive drug would be the drug that is performing the balance between a sufficient immunosuppression to inhibit any damage to the graft by the immune response that is trying to react to the transplanted graft, and it will be low enough to minimize and have as little as possible side effects, and leave a valid immune response that can fight infections, all kinds of infections, whether viral, bacterial, or fungal. So this kind of balance is really not an easy task. But I think what we learned over the past years all together as in the transplant community is how to make this balance, and I think we have a good experience in this now. In the old days, the immunosuppression in front of you, which is the corticosteroids and the azathioprine in the 60s, were the only available immunosuppression. And if they worked well, or more or less, they managed the kidney world, in the liver world, actually the 60s and the 70s, we had very dismal results in the term of 40 to 50% success rates. In the best hands, whether if we speak in England, in the Cambridge by Sir Roy Kall, or the pop of liver transplantation, Thomas Starzl, where he started in Denver, Colorado, or later on in the different centers until Pittsburgh, the results they had, which were really the best results in that time, were about 50 to 60% only, which was really not very nice results. And it was until actually it's not 1983. Actually, it was until 1978 or 79 when Sir Roy Kall from Cambridge pioneered this first CNI, the cyclosporin. It was later on approved by the FDA. Maybe it took some time, but this was a breakthrough in liver transplantation. We all witnessed that the results. There have been actually 3 breakthroughs in the history of liver transplantation. The first one was the appearance of the cyclosporin drug, and the post-cyclosporin era in the early 80s, and thereafter we started to witness some good results like 70% and 80% and really survival rates and graft survival, whether overall survival or graft survival, really getting better and better with time. In terms of timeline, as I told you, in 1983, the cyclosporin made the breakthrough, and then in 1994, pioneered by Professor Thomas Starzl in Pittsburgh, Pennsylvania, where the tacrolimus were started for the very first time, although maybe it was coming in from Japan, but there was the real work, and they tried it in all organs, in the heart, in the liver, in the kidney, and later on, 1995, the MMF occurred. 1998, the basileximab for the kidney, followed by the sirolimus, the first mTORs in 2001, and the late 2009-10, the everolimus that we are started to use later on in the liver world as well. So if you look to this timeline, it stopped at 2012. Some 8 to 9 years ago, where no more new immunosuppressive drug enter our armamentarium, and actually we are stuck with this. But I must say that the CNI, whether it's the cyclosporin or the tacrolimus, really played an important role in ameliorating the results and getting much better results than thereafter. I think that my friend Professor Effat Kamil will take the thread from me, starting from here. But maybe if I just want to go to this slide, if you want to start by it again, now in the liver world, although we use both the cyclosporin and the tacrolimus over the past 20 years, I would say 80% we're using tacrolimus rather than cyclosporin because of many papers and many studies that potentiated the results of tacrolimus as a better immunosuppression and it's a more potent immunosuppression than cyclosporin. And I think Professor Effat will elaborate more on this. So the floor is yours, Effat. Thank you.
I would like to start by thanking my dear friend, Professor Mahmoud El Meteini, for his nice presentation. I'd like to thank Dr. Sameh El-Bagouri, the country head of Sandoz Egypt, and Sandoz for their kind invitation. I would like also to thank the coordinators of this meeting, Dr. Mohammed Hamid, the therapeutic area manager of Sandoz, and Dr. Mariam Fouad, the medical advisor of the immunology and transplantation. And I would like to really welcome our panelists, our very distinguished panelists. We're really honored to be part of this meeting tonight. Dr. Mahmoud actually did a very nice introduction. Let me continue with the slides. Somehow I can't, yes. Well, actually, I totally agree that tacrolimus has been the mainstay in living donor liver transplantation and in liver transplantation in general for obvious improvement of the results. They have been introduced, tacrolimus and MMF, in 1995, and they have significantly lowered the rates of acute rejection. And as you can see here, the acute rejection rate has gone down from 60% to about 20%, and the 1-year survival, graft survival, has increased from 50% to about 90%. We all know that acute rejection, although the name is very scary, it's actually not a big problem in management. However, it involves giving large doses of steroids, and if you have repeated attacks of acute rejection, you start going into infective complications and other complications. So it's really important to have the acute rejection episodes under control. Next, please. Next, please. You can see that tacrolimus has definitely reduced the number of acute rejections, and I have just said more acute rejections, more steroids, and more infection rates. So definitely this has been an advantage. And if we can talk about the milestones of organ transplantation, liver transplantation, cyclosporin has been the main milestone in the development of liver transplantation as a therapeutic option in the management of end-stage liver disease. But ProGraf or tacrolimus have really moved liver transplantation into a much safer procedure. Next, please. The long-term efficacy of tacrolimus has been reflected in both patient survival and graft survival, as you can see from those graphs, with the years post-liver transplantation. And this is important because what we look at now is not only the short-term survival after liver transplantation, but we look more for the long-term survival after transplantation. And this is the main challenge, really, in organ transplantation and in liver transplantation in particular. The acute problems related to liver transplantation have sort of been sorted out, most of them, I would say, but we still have a mortality, but definitely our biggest challenge is in the long-term survival. Next, please. If you see how things are today, or our status quo, you would say that prevention of acute rejection has greatly improved. Minimization will always be a challenge, and we talk about side effects of immunosuppression. There are new drugs in the market trying to push this. I think the key is in managing immunosuppression doses and in minimizing as much as possible and in weaning whenever this is possible and promoting long-term survival. And of course, something that is always of concern is to be cost effective, since these are treatments for lifetime. The challenges meeting us in the 21st century, and this is very important actually, is increased demand, which will never stop, for organ donors. Extended donor criteria are used around the world. Our solution has been living, as my friend Dr. Mahmoud said, we would have liked to see cadaveric transplantation, because without cadaveric transplantation, you can't really cover all your needs. And even in the countries where there is cadaveric transplantation, the organ needs are not fully satisfied. And that's why they've extended their donor criteria. Of course, as your experience goes ahead, you involve sicker patients with higher MELD score, and these are definitely a challenge by themselves. I think both our groups have managed to reach a long way in the experience and in dealing with the sick. I must say that because of not having cadaveric transplantation, we don't take the very sick, really sick patients, the ICU patients in very bad conditions. You need more of a cadaveric setup in this situation. Metabolic disorders, of course, metabolic disorders, and specifically NASH is the new epidemic of the 21st century. As you have seen, HCV is being under control with the new DAAs, and it's no more really the upcoming challenge as when we started transplantation. And now we see more and more patients with NASH, and this is the new norm. And also we see a lot of patients with hepatocellular carcinoma. I think in our centers and in most centers around the world, more than 25 to 30% of our patients are HCC patients. And this is becoming the main indication for living donor liver transplantation in our part of the world. And this is, of course, as a sequence to HCV and HPV. Renal toxicity remains a challenge, and there are new drugs that have really sorted out this problem that can be given as a loading drug, and then you can introduce the CNIs at a much later stage, saving the kidney. Next, please. I would conclude by saying that over the past 19 years, Egypt has had a pivotal role in liver transplantation across the region. Tacrolimus is the backbone of immunosuppression in the field of liver transplantation with long-term efficiency. Tacrolimus, MMF, and corticosteroids among the frequently used immunosuppression in liver transplantation. I'd like to thank you, and we should proceed with the next of the session.
Thank you, Effat. I think both of us have now set the stage very nicely for coming next. And I think according to the agenda, I don't know if it's up to me to present Dr. Rita, or you?
Allow me, Dr. Mahmoud, I will start by introducing Dr. Peter Effat, who is the head of the medical affairs of Sandoz. He has a small presentation to give to set further the floor for Dr. Rita's presentation.
اوكي. مساء الخير على حضراتكم جميعا. في البدايه طبعا بحب ارحب بكل البارتيسبنتس اللي موجودين معانا النهارده، وطبعا سبيشال ثانكس لاساتذتنا الكبار اللي بيشاركونا النهارده، الاستاذ الدكتور محمود المتيني والاستاذ الدكتور رفعت كامل، اللي احنا بنشكرهم على يعني هم تفضلوا مشكورين بكرامهم ان هم يكونوا جزء النهارده من الفيري سبيشال ايفنت بتاعنا. هو اكشولي اتس ريلي فيري سبيشال انه اول مره يتعمل فيرتشوال لونش ان ايجيبت، فان شاء الله يا رب نعدي الازمه دي ونشوف حضراتكم باذن الله فيس تو فيس. الحاجه الثانيه ان احنا بنقدم يمكن زي ما تفضلوا اساتذتنا عرضوا في السلايدز اللي فاتت، وبعد ما نشوف النهارده ان شاء الله باقي اليوم ان احنا بنقدم البورتفوليو بتاع ترانسبلانتيشن اميونوسبريسنت، سواء التكرولمس او المايكوفينوليت، ادويه اسينشال از immunosuppressants. اسمحوا لي في الدقائق اللي جايه انا هدي لحضراتكم بريفنج سريع على شركه ساندوز عموما. فشركه ساندوز يمكن برضو بنحب نعرف حضراتكم انه شركه نوفارتس العالميه، نوفارتس الكبيره، هي بتتكون من 3 ديفيجنز. شركه ساندوز هي الاندستري ليدنج ديفيجن مسؤوله عن كاتيجوري معينه من الادويه، هي البايوسيميلرز والاوف بيتنت ميديسنز. ده بالاضافه للاذر 2 ديفيجنز اللي الثلاثه مع بعض بيكونوا شركه نوفارتس، اللي هي نوفارتس اونكولوجي ونوفارتس فارماسيوتيك. يمكن سريعا جدا برضو about ساندوز. شركه ساندوز is currently operating في اكثر من 160 دوله world wide، وبتعتبر جلوبال ريدر اند رانكد نمبر 1 في 3 ثيرابي ارياز. الثيرابي اريا الاولانيه هي الاريا بتاعت البيولوجيكس او البايوسيميلرز بحوالي 8 ماركتد برودكتس وفيري سترونج اند فيري بروميسنج جايد لاينز، رينج كبير من الادويه for the treatment of different types of diseases starting from الجروث هرمون deficiency diseases, blood cancers، رينج كبير من الاوتوميون diseases، IBS، IBD، ومجموعه كبيره جدا من الادويه. الثيرابي اريا الثانيه اللي شركه ساندوز از ليدنج فيها جلوبالي هي الانتي انفكتيفز. يمكن شركه ساندوز از جلوبال ليدر اوف انتي، ومش بس كده لكن كمان ده بهيستوري بيرجع لل50s. في بدايه الخمسينات شركه ساندوز كانت هي اول شركه قدرت تقدم الاورال بنسلين. ثالث ثيرابي اريا هي الانكولوجي انجكتبلز، الجينيريك انكولوجي. شركه ساندوز از جلوبال ليدر باكثر من 50 برودكت في الاريا بتاعت الانكولوجي، برضه رينج كبير ستارتنج من الكيموثيرابي مرورا بالهرمونال ثيرابي، سبورتيف كير، ووصولا للبيولوجيكس. يمكن برضو احنا شركه ساندوز بتتميز ان البورتفوليو بتاع الادويه از فيري جود وان، يعني احنا بنتميز بحوالي اكثر من 1000 موليكيول. aside from البايوسيميلرز والانتي انفكتيفز والانكولوجي اللي اتكلمنا عليهم، احنا كمان اوبريتنج في الريسبيراتوري، CVS، CMS، وترانسبلانتيشن واذرز. يمكن من حوالي 3 سنين احنا كان عدد العيانين اللي احنا قدرنا نوصل لهم ونخدمهم بالادويه بتاعتنا بس كانوا اكسيدنج ال 500 مليون بيشنت وورلد وايد. يمكن دول بيمثلوا اكتر من نص البيشنت اللي شركه نوفارتس عموما بتقدر توصل لهم وتخدمهم بادويتها. احنا كنا زي ما بقول لحضراتكم من 3 سنين شركه نوفارتس عموما بتوصل ل 1 بليون بيشنت وورلد وايد. احنا از ساندوز كنا بنمثل حوالي اكتر من 500 مليون بيشنت، اند اور اسبيريشن ان احنا في سنين قليله نقدر نخدم بادويتنا اكتر من 1 بليون بيشنت. طبعا it goes without saying، لا خلاف على ان المودرن ميديسنز has changed the world، بس unfortunately هي ما قدرتش تحل مشكله الاكسس. يمكن ال 100 سنه اللي فاتوا العلم شهد ان احنا حصل هيوج ميديكال بريكثروز، كان في ستارتنج من الديسكفري بتاعه البنسلين ووصولا للكاتنج ايدج الكانسر ميديكيشنز. despite of this huge development اللي حصلت still في اكتر من 2 بليون بيشنت is lacking access للاسنشال ميديكيشنز اللي هم محتاجينها. يمكن هنا بقى بيجي البيوتي في الاوفرنج بتاعنا، طبعا مع الاخذ في الاعتبار زي ما حضراتكم عارفين انه البريشر اللي حاصل على الهيلث كير از ستيدلي انكريزنج، والديماند بيزيد سنه عن سنه نتيجه طبعا ان الجلوبال بوبيوليشن نفسها از استيميتد ان هي بحلول سنه 2030 هتوصل ل 8.5 بليون. مش بس كده لكن كمان الايجنج بوبيوليشن في خلال 10 سنين هيكون عندنا اكثر من 1.5 بليون معديين ال 60 سنه، والهيلث كير اكسبنديتشر في ال5 سنين الجايين يستمر ان هو يوصل ل 13 تريليون يو اس دولار. ده بيخلق هيوج بريشر على الهيلث كير سيستمز عموما ان هي تقدر توفي الديماند العالي جدا ده. ذاتس واي احنا بنقول انه الجينيركس ميديسنز ار كي عشان السستينابيلتي بتاعت الهيلث كير سيستم. يمكن على سبيل المثال في خلال من سنه 2005 ل 2015 في اوروبا ضعف عدد العيانين قدروا يوصلوا للادويه اللي هم يستحقوها طبعا نتيجه وجود الاوف بيتنت ميديسنز والجينيركس ميديسنز اللي بتقدر تفيل الجاب دي وتقدر تفيل النيد كبيره في الجزئيه. اخيرا اخر سلايد ليا هقولها او اي وود لايك تو شير مع حضراتكم ان احنا في ساندوز اور بيربس ان احنا تو بايونير اكسس للبيشنتس دول عن طريق ان احنا بنشتغل in close collaboration مع الساينتفك بارتنرز بتوعنا to maximize our work to make access happen، ان احنا نخلي العيانين فعلا يقدر يوصل لهم الادويه اللي هي البالانسنج الفورمولا اللي هي ما بين we are offering ادويه that is really affordable and at the same time it's multinational standard, it's quality products لا تقل تماما عن الريفرنس برودكتس. بلس ان احنا we are working ان احنا نقلل البيردن اللي محطوط على الهيلث كير سيستمز. احنا بندي simply more spaces. احنا we are trying to spare البادجتس بتاعت الهيلث كير سيستمز دي عشان تقدر تغطي عدد اكبر وتوصل عدد اكبر من العيانين، بس مش بس كده كمان، لكن كمان to spare their budgets عشان تقدر توصل كمان ان هي تغطي other essential medications. Thank you very much again. نتمنى يا رب ان احنا قريب نشوف حضراتكم in face to face. Thank you so much لاساتذتنا ولكل البارتيسيبانتس.
Thank you very much, Dr. Peter. I think this is a very important subject, actually, because we all know, especially in a country like ours, that the cost of treatment is very high, and anything that would give quality treatments together with cutting the cost is definitely most welcome in a country in this part of the world. So thank you very much for this economics, because the economics of treatment are just as important as the efficiency and the pharmacogenetics of the treatment. I'll leave now my dear friend, Professor Meteini, to present our main speaker, Dr. Rita.
Thank you, Effat. Thank you very much, Peter. I really enjoyed your talk. Very informative, very concise to the point. And I would like, if you don't mind, to share these slides with you, because now being president of the university, في جزء سياسي في الموضوع.
With pleasure.
Now it gives me great pleasure to Rita. Are you there, Rita? Can you hear me? Okay. It gives me great pleasure to introduce our guest speaker today, Professor Rita Allowy from Cincinnati, United States of America. Actually, her bio is very impressive, and I've been really impressed by the data in it. She's a research professor in the College of Medicine at the University of Cincinnati and the director of transplant clinical research. She received her Doctor of Pharmacy degree from the University of Tennessee College of Pharmacy in 1988. She practiced multiple areas, actually, including kidney, liver, and pancreas, solid organ transplantation. And her work in transplantation pharmacotherapy has generated numerous publications and opportunities to present specific research results and overview of transplant immunosuppression. Dr. Allowy individual research efforts focus on the goal of individualizing immunosuppressive regimen in the transplant patient. She currently directs several industry-sponsored and investigator-initiated investigational drug trials, of which she served as both principal and collaborating investigator. She's an elected member of the American College of Clinical Pharmacy, the American Society of Transplantation, and also a member of the Immunology Transplant Practice Research Network. She served as the president of the Mid-South College of Clinical Pharmacy and the ACCP Transplant Immunology PRN Chair. As a member of the AST, she previously served as a counselor at large on the AST board, chair of Transplant Pharmacy Community of Practice, and member of the Public Policy and Governance Committees. Her long-standing mentoring and training efforts in transplantation were recognized by the AST when she was awarded the 2013 AST Mentoring Award. Dr. Rita, it's a pleasure to have you with us, and the floor is yours, and we are really very eager to listen from you. Thank you very much.
Thank you very much for that kind introduction. I have a very special connection to Egypt. I would not be where I am today and enjoyed the career that I have in transplantation had it not been for Dr. Osama Gaber, who is actually from Cairo, Egypt as well. He was my mentor, and he at the University of Tennessee, and was the first one to introduce me to transplantation. And I'm truly honored to have been associated with him, and I feel very honored again to be here to present this information to you today. So the title of the presentation today is Generics, Good Medicine or Cheap Fix, and we're going to focus on the data in liver transplant recipients. So as of disclosure, I've received several clinical research grants. I serve on the advisory board for Veloxis and Sanofi and the Speakers Bureau for Sanofi and Veloxis. I was provided honoraria to present this talk today, and this presentation does not include discussion of off-label or investigational use drugs. The objectives today are to identify the pharmacokinetic parameters that are related, that are used to evaluate, okay, I'll just press on. The objectives today are to identify the pharmacokinetic parameters which are evaluated to determine bioequivalence in healthy volunteer studies. And we'll come to this in more discussion later on, but as you may be aware, the testing is only required in healthy volunteers for a generic product to be marketed, and we'll cover that more later. Next is to review the current bioequivalence data with generic immunosuppressants, not in healthy volunteers, but in actual stable liver transplant recipients. And then last, identify the impact of the adoption of generic tacrolimus on the United States transplant recipients. So I need to really start with a definition of bioequivalence. And this is the regulatory definition of bioequivalence, and then I'll break it down further. But essentially, it's the absence of a significant difference in the rate and extent of which an active ingredient becomes available at the site of action during similar conditions in a well-designed study. So the bottom line is, for you to accept generics or bioequivalence testing, you have to accept the fact that if the active ingredient, such as tacrolimus, makes it to the bloodstream at a similar rate and extent without a significant difference, we would have the ultimate same biologic impact. And that's really the assumptions that you make going forward with bioequivalence testing. From a historical perspective in the United States, the FDA approval process has been in place for over 30 years. The Egypt Ministry of Health reviews the FDA and the EMA bioequivalence data for approval. The process of this is still heavily debated and is fueled when generic equivalents for cornerstone treatments such as tacrolimus are introduced into new therapeutic areas, especially therapeutic areas such as transplantation that are life-saving. The debate is fueled actually by a lack of understanding of the generic approval process, which I'm hopefully will be able to provide you some insight to today. The FDA and the EMA staunchly defend their standards, and the brand manufacturers attempt to discredit this process. In the United States in 2012, the Generic Users Drug Fee Act was passed. And what this resulted in was when generic drug companies presented their applications for new generic products, the FDA was able to charge an additional fee. And the money from this fee would go to additional studies from generic products outside of those within healthy volunteers. So just in brief, generic products must be the same active ingredient. And in this case, we're referring to tacrolimus. Now, while there may be several dosage forms of tacrolimus, there are actually very few manufacturers of the tacrolimus raw material, and that's important to understand. The dosage forms must be the same, the route of administration must be the same, and the strength and the concentrations of which the generic formulations are made must be the same. So, for example, in the case of tacrolimus, we would be able to have a 0.5, a 1 milligram, or a 5 milligram strength. But for example, a generic manufacturer would not be able to market, say, a 2.5 or a 3 milligram strength. However, the generic products may be different in regards to shape, scoring, release mechanisms, excipients, packaging, and expiration times. Now, the standard design of bioequivalent studies, and it is really the root of a lot of concerns that are related to these tests. They are two-way crossover studies, and they really only have to be 24 or 36 healthy adults. There is only a single oral dose of the generic product and the brand product administered to the healthy adults, and the pharmacokinetic parameters are calculated from the time concentration curves. So this reviews the process. What appears to be to most transplanters or to any other clinical professional, only 24 to 36 healthy volunteers may be required to do a bioequivalent study. They can go higher, they can enroll in numbers, but what is an interesting understanding is really the higher the number of patients that are enrolled in a study, the limitation it has on the variability of a product. So it's really quite good to see a study that enrolls as low a number of patients of 24 to 36 and still be able to pass the bioequivalence testing parameters from a statistical perspective. Those patients are randomized to receive one dose, the same milligram of brand, and 1 single milligram of the generic dose. Once those doses are administered, several blood samples are taken from the patient and are used to plot and construct this time concentration curve that you see depicted here. After a significant washout period, the healthy volunteers are converted to the opposite brand or generic with the exact same dose, and the exact same blood concentrations are taken, and a replicate time concentration curve is formed. Now, from that, we compare these pharmacokinetic values, the mean Cmax for the rate of absorption and the mean AUC to classify the extent of absorption. And it's quite interesting. Most transplant clinicians are pretty good pharmacokineticists, and they have a good understanding of this. We all are familiar with the concentration max that occurs at the peak, and the time of which it occurs is the time of the maximum concentration. However, what is very important is the assessment of the area under the curve, or the extent of exposure of a drug. And essentially, the area under the curve is just a calculation of this area to represent the overall exposure of the dosage form that was administered. So the FDA bioequivalence standards include 2 one-sided 90% confidence procedures that result in the appreciation that there will be a difference in the mean values that you observe, but provide reasonable assurance that these mean treatment differences are acceptable. The statistical procedures that are implied within the guidance recommendations are confidence intervals between 80 to 125% for the AUC by the FDA. The EMA has actually chosen to narrow those confidence intervals to 90 to 111% for AUC for narrow therapeutic index drugs, of which tacrolimus is one. And also the concentration max confidence interval should fall between 80 and 125% for log transform data. Now, you may be led to believe that this could result in there being a total of a 45% difference in the AUC, but I hope I'm able to show you with how the calculations work that is actually not the case. So we began the experiment in the United States that you guys are embarking on in Egypt now, back in the early 2000s. And there was significant public concern regarding the immunosuppressants. So essentially, the main concern stemmed from the fact that standard bioequivalence approval process based upon the two-way crossover design in healthy volunteers or healthy individuals I just described was really not effective and not a valid approach for approval of generic immunosuppressants in transplant patients. The concern was for a lack of data in transplant recipients, especially when you consider this a high-risk procedure and you think of the impact of poor absorption based upon the genetic differences, especially in the liver transplant population, based upon the genetic metabolism of tacrolimus, both in the gut and the liver. Also, the concern was based upon the need to recognize tacrolimus as a narrow therapeutic index drug, therefore implementing more strict bioequivalence standards. And it was agreed upon by the FDA and the EMA that tacrolimus did fall into the criteria of a narrow therapeutic index drug. And also there was a concern, for a lack of comparison for bioequivalence among the various generic products. And for example, when a generic is approved for the market, the generic is compared to the brand name. However, multiple generics are not required to be tested against each other. However, in day-to-day practice, once multiple generic formulations infiltrate the marketplace, there will be quite a bit of formulation switching if you choose to do that. There are several consensus documents that have been developed by professional societies in Europe, US, and in Canada that cautioned against the use of generic immunosuppressants. But in 2020, I want to point you to the dates of these publications: 2003, 2003, 2012, 2011, and 2009. So the people that try to discredit generic pharmaceutical agents really want to convince you that bioequivalence testing is vilified and it is not appropriate. However, bioequivalence testing is used throughout the industry anytime a new dosage form is created or there is a change in the manufacturing process. Healthy volunteer studies with single dose undergo bioequivalence to approve the new formulations. And I want to give you an example. We all remember that Sandoz oral solution was the product that was approved in the phase 3 trials. However, when we were provided Sandoz capsules, we were provided Sandoz capsules after a bioequivalence study. Same way with the phase 3 studies with the oral solution when we went to the oral capsules. Same with mycophenolate mofetil. The phase 3 studies were with 250 milligram capsules, and we have a variety of CellCept dosage forms nowadays that were provided to us via bioequivalence testing. Prograf, if you remember in the phase 3 trials, were conducted with 1 milligram capsules, and now through bioequivalence studies, we have 5 milligram capsules as well. Now, what is interesting to denote is that the phase 3 trials of sirolimus utilized Rapamune oral solution of 1 milligram per ml. As they began to reach approval for this product, they attempted to develop the Rapamune 1 milligram oral dosage form, and they were unfortunately not able to develop a bioequivalent 1 milligram tablet for Rapamune. So they had to go back and repeat some of their phase 3 studies with a tablet formulation to obtain FDA approval of the tablet. So bioequivalence testing is used throughout the pharmaceutical industry, not just for generic drug approval. So this takes me into the immunosuppressant generic summary as has happened in the United States. In 1998, the first modified release cyclosporin liquid generic formulation was marketed, and it was approved at that time for use within water, orange juice, milk, soda, etc. But it was later withdrawn from the market because it was found to not be bioequivalent when mixed with apple juice. So it was removed from the market. In 1999, the first modified release cyclosporin tablet generic was marketed. And interestingly, in 2009, there was a major shift in the United States. Initially, mycophenolate mofetil generic was marketed in 2009, and within 2 to 3 months, the first generic tacrolimus product was marketed. And by 2013, there was already a greater than 50% infiltration of the marketplace with generics, of which I will show you more data later on in the presentation. Since that time, in 2014, we have a sirolimus generic, and in 2019, the first everolimus generic was approved. I'm unsure if it is marketed yet or not. So if you remember back, I told you that the FDA created the generic users drug fee act, and that freed up funds to do additional studies in generic products. And so the FDA put out an RFA to look at the pharmacokinetic studies of generic tacrolimus in transplant patients. And if you look in the funding opportunities below that circled in red, you can see that the goal of this proposal was to compare the pharmacokinetics of 2 sources of generic tacrolimus, not just one, but 2 sources of generic tacrolimus, to Prograf in stable transplant patients. They wanted the transplant patients to be switched between both brand and generic, and between generic and brand. And they wanted the outcomes of the study to help address the public concerns that were highlighted previously regarding the quality of generic tacrolimus and improve the review practices of generic tacrolimus formulations if necessary. So me and my team submitted the RFA proposal, and on September of 2012, we were awarded the proposal. In October of 2012, we went to the FDA, and they assembled a group of representation from Transplantation to review the scientific design of the protocol. And you can see the representation from the FDA listed here, from the American Society of Transplant, Robert Gaston, Rosalind Mannon, and David Cohen. From the American Society of Transplant Surgery, Jorn Klintbaum, Art Matus, Mitch Henry, and Kim Altoff. And then the grant awardees, myself, Uwe Christians, and Alexander Vinks. And the result of this meeting, there was a unanimous consensus on the protocol design as was submitted. So therefore, no changes were made. So to give you a brief overview of this design, it was a fully replicated, blinded, 6-period crossover study in 2 centers. We utilized a stable adult liver transplant population on a twice-daily formulation of Prograf, and we excluded any patients that may be on drugs that were known to interfere with tacrolimus metabolism or pharmacokinetics. Our intervention was to identify, based upon the healthy volunteer data that we had, which was all that we had from all the different generic formulations, but we attempted to identify what we thought would be the 2 most disparate, the 2 most different tacrolimus generics among those that were currently approved in the United States, and identify one as generic high and one as generic low. And in this study design, we actually did 6 pharmacokinetic assessments over a 12-hour period, where we administered the brand product twice, generic high product twice, and the generic low product twice. And then the resulting pharmacokinetic parameters were assessed for each pharmacokinetic visit. We compared, again, generic versus generics as required by the RFA to the Prograf, but also the 2 generics to themselves, and a variety of other outcomes of interest that I'll be sharing with you as we go along. So again, to reflect on our protocol design, it was a 6-period crossover study. We were able to measure tacrolimus and its metabolites via the most sophisticated LCMS and analytical methods. We also genotyped the recipients to ascertain if there were any differences in the metabolism based upon formulation by the genetic phenotypes of the recipients. And if you look here, the patients were screened and then brought in for randomization. And they were randomized to go into one of 3 sequences that were generated by the computer to administer. Yes? To administer these products over a 6-week period. And what you can see again, brand administered twice in the same patient, we were able to compare the within subject variability that occurred between brand, between generic high, and also between generic low. And that goes to show you that when you measure a tacrolimus level, even in the same patient on the same dose, you're going to get a slightly different level upon return. However, after a week of administration of these products, we conducted a 12-hour pharmacokinetic parameter with the morning dose over 6 full PK periods. The blinding method, we initially attempted to discuss over-encapsulation of the formulation so that we would not be able to see which formulation we were giving. However, anytime you do over-encapsulation, there's a risk that the dissolution of what you use to encapsulate the product will impact the pharmacokinetic properties. So we decided against that, and we went to the following blinding criteria. So at our site, we conducted the pharmacokinetic studies, and the subjects were randomly assigned to study drug sequence based upon what the investigational drug pharmacy gave us. and the sequence of this drug administration was unknown to the study staff. So we could see in our hands which product we gave the patient, but we did not know which product we would give them from week to week to week. Once we collected these samples, we sent them to the analytical site for analysis by mass spectrometry, and those samples were blinded. The samples were shipped and only identified by subject number, date, and time of samples. The levels were quantified, and the results were then provided to the data management team and merged into a database. A separate pharmacokinetic analysis team basically took those levels and calculated all of the pharmacokinetic parameters that I described to you before and that I'll present to you as we follow in an unblinded fashion. After all the pharmacokinetic parameters were calculated, the IDS pharmacy provided the pharmacokinetic analysis team with the unblinding of the results, and they were able to assess the formulation impact. And so for just fun today, I'm going to keep the audience blinded to the formulation until the conclusion of the talk. So first, when it came down to product selection, at this time there were 5 generic tacrolimus products available in the United States. When we looked to select the 2 most disparate products, the predicted generic high product was clearly identified based upon our selection criteria. So there was really no question what that product would be. When it came to the generic low candidate, we identified 2 candidates for that, candidate number 1 and candidate number 2. In these type of studies, it's very, very important that you obtain a quality independent of the manufacturer of the same lot number. And we were unable to get a significant quantity to complete the study of candidate 1. So we moved on to candidate 2, of which we could get a significant quantity of the same lot through the wholesaler, and we weren't, highlighting to the manufacturer that we were doing these additional tests. So in any type of pharmacokinetic study, and as we all know in transplantation, adherence is very important. So we went to great lengths to monitor the adherence in these patients. So the patients were administered their dose of tacrolimus in pill bottles that included a cap that had a computer chip in the cap that would let us know when they took the cap off and when they put the cap back on. In theory, that was a surrogate for when they actually took their medicine. We don't know that they actually put it in their mouth at that time, but that's what we instructed them to do. And you can see that there was a very high rate of adherence in all the patient populations. In addition to that, when the patients came in each week, we reviewed a patient diary that they documented by hand, and we also counted the pills that we had dispensed from the previous week. I'm not going to present the data today, but an additional adherence marker was we collected via a finger stick or a prick and applied that blood to a protein saver card, and we used those dry blood spots to analyze the tacrolimus level on a daily basis. However, that was not available to us at the time of the pharmacokinetic study. We evaluated it later. So per protocol, patients were excluded from pharmacokinetic analysis if they missed a dose within 48 hours of the PK visit, as evidenced by the men's cap, the pill count, or the patient diary. And you have to understand that for the patient to be evaluated, they had to complete all 6 of the pharmacokinetic visits, or we couldn't make all the comparisons that we needed to make. So this is a busy slide, but I'll briefly review it and just say that we assessed eligibility in 337 patients and excluded 297 for a variety of reasons. We randomized 40. 39 were actually allocated to the intervention. 1 did not allocate to the intervention because they missed their baseline visit. In terms of follow-up, there were 2 patients that were discontinued from the intervention. 1 was unable to complete pharmacokinetic studies over the 6-week period due to personal reasons, and 1 patient missed several doses of their tacrolimus product before their first PK visit. We ended up analyzing of that 36, and one, again, we had to exclude from non-adherence within 48 hours of the PK visit. So based upon that, we ended up with 36 subjects for analysis. It represented our center very well. Median age of 57, 50%, 47% male gender, a low African-American percentage. Unlike your program in Egypt, we have at this time 100% deceased donors with no living donors. The time post-transplant was roughly 3 years. 30% of the patients had diabetes. 8% were on steroids. 89% were on CellCept. And the milligram per day dose of tacrolimus in the stable patients 3 years out was 2 milligrams twice a day. So let me take a moment to describe to you the concept of scaled average bioequivalence. This is a more stringent evaluation of bioequivalence than the confidence interval testing. And what this bioequivalent metric does is it allows for narrow therapeutic index drugs to be approved under a more narrow confidence interval based upon what the reference product or the brand name products within subject variability is. Now, I'll take you through this. So if you're within subject variability is roughly 22% or so, you can see that the barriers of the confidence interval remains the same, 80 to 125. If by chance you're within subject variability is less than 22% or so, you can see that actually the confidence intervals narrow and make it more difficult to achieve the bioequivalence testing limits. In regards to the 30%, in general, from an FDA perspective, drugs within subject variability of greater than 30% are considered highly variable. So I want to take a minute to orient you to this slide. So this is the classic tacrolimus time concentration curve that we're used to seeing with time across the x-axis and tacrolimus concentration across the y-axis. Each of these open dots represent a time in which we tested a tacrolimus blood sample. You can see the sample times here, time 0, 20, 40, 60 minutes, 80, 100, 120 minutes, 140, 160, 180 minutes, and then 4, 5, 6, 8, and 12 hours. And all of our patients fasted until 4 hours after the morning draw. And the purpose for that was because the peak or the C maximum concentration is really the concentration that drives your area under the curve, we wanted to make sure that we didn't have any interference with this. And you can see when you go out to 4 hours, we have certainly passed the time of peak absorption for tacrolimus formulation. So I told you that we had 3 products that we administered 2 times. So this graphic depiction is when in liver transplant recipients, we administered product 1 the first time. This is when we administered product 1 the second time. Same dose, same patients. This is product 2 the first time. This is product 2 the second time. This is product 3 the first time. This is product 3 the second time. And this is just a graphic depiction of all the products overlaid over themselves. And you can see by looking at the mean values and the standard error of the mean bars that there was no statistically difference between formulations identified at any time point. And even when you start to see differences, which you may identify just by visual inspection here, we're talking about a level of like 9.8 compared to 12 or so. All right. So I will again provide you with the pharmacokinetic parameters that resulted from these calculations, from these graphs. So in the liver population, the area under the curve was 62 for brand, 65 for generic high, and 66 for generic low. The concentration max was 10.4 for brand, 11.3 for generic high, and 11.68 for generic low. The concentration min was 3.2 versus 3.5 versus 3.4. And the time to maximum concentration was 1.3 versus 1.2 versus 1.2 hours post-dose. I'm going to take a little bit of time to explain this slide because it's going to be replicated in future in subsequent analyses of the pharmacokinetic parameters. So this looks at tacrolimus AUC for scaled average bioequivalence in the liver. Okay, so when product 1 is the reference product, so when we compare product 1 to itself, so the within subject variability of product 1 for AUC is 14%. When we compare the within subject variability for the reference product when it was product 2 or one of the generics, it was 14.2. When we compare the within product variability when it's with product 3, which was a generic product, it was 12.7% within subject variability. Now, I think that this should strike all of us. I think that I've written pharmacokinetic papers all my life where I've said that tacrolimus is a high intra and inter patient variable drug. And I think that we make those assumptions based upon what we see clinically when the effects such as diet, time of dose, time of level collection, etc., are incorporated into our observations. However, in a very controlled environment, you can actually see that the within subject variability is really actually low for tacrolimus products. So that's an interesting observation, and it kind of changes the way we think in some cases. But let's go back to the basics. So for area under the curve, when we compared product 1 to product 2, we observed the mean ratio of the AUC of 104, which means that product 1 was 4.8% higher than product 2. In this comparison, product 1 was 6.3% higher than product 3. In this comparison, product 2 was roughly 1.5% lower than product 3. And in this comparison, product 3 was 1.5% higher than product 2. So when we look at these within subject variability calculations or observations, we then calculate a scaled bioequivalence limit. And you remember before that I told you that based upon these within subject variability numbers, if they're less than 22 or so, these numbers will actually narrow to become less than 85 to 120 or 90 to 111 that you see being required for average bioequivalence. So this was the scaled average bioequivalence that was calculated. So this was what was calculated by our parameters, that if the observed confidence interval fell within these ranges, it would be considered bioequivalent by both criteria. So when we observe the 90% confidence interval, you can see what was observed here falls within the range of the scaled average bioequivalence limit for comparison of all of these products. And just by observation, you can see that this falls between the 90 to 111 more stringent criteria of the EU, even in transplant patients. And based upon the FDA approval product standards and also the EU product standards for area under the curve assessment, these formulations all passed bioequivalence testing parameters. So I'll briefly now move more quickly through Cmax. I think we all know that Cmax is obviously more variable because it requires you to make absolutely sure that you're able to measure when the Cmax occurs in all the patients. And you can see that within subject variability between product 1 was 18%. Within subject variability of product 2 was 24%. And within product variability for product 3 was 22%. And subsequent to that, based upon that within subject variability, you see the scaled average limits for these two are narrowed, but you can see here when they within subject variability goes greater than 22%, the scaled average bioequivalence will not widen any further. It will stop at 80 to 125%. When you look at the 90% observed confidence interval, it again falls within the scaled average bioequivalence for all of these product comparisons. And again, it passed the FDA and AMA approval criteria. For the C minimum concentrations, and keep in mind that these are minimum concentrations from the morning dose. We typically clinically monitor the minimum or the trough concentrations from the nighttime dose. But even when we looked at the C min concentrations, you can see that the within subject variability, again, was back lower than the C max. We were able to calculate the scaled average bioequivalence limit, observe and see what our observed confidence limits were. And again, they fell within the confidence limit testing. So in terms of adverse effects by formulation, we would not expect it, but in this short study, there was no acute rejection. Graft survival and patient survival was 100%. But as we're all familiar, all of our transplant patients experience a series of adverse events, and 69% of the liver transplant patients reported an adverse event. They're divided here by group, and when you compare them by product 1, product 2, and product 3, there's no statistical differences, but you can see slight trends. There were higher CNS reports in the liver population compared to that of the kidney, but really we didn't see, in gastrointestinal toxicities or any of the other toxicities, major differences in the reported adverse events by the patients. So to understand the pharmacokinetics and bioequivalence testing, you really have to appreciate the complexity of tacrolimus metabolism and its impact on liver transplants, which is quite unusual, as I'm sure you're aware of. So Adport, when it's administered to a patient and goes by mouth inside the GI tract, it's transported across the gut membrane by gut transport proteins. It may be exported back into the GI tract for elimination. It may also be absorbed within the gut wall of where there's significant gut cytochrome P450 metabolizing enzymes that are located there. So there is this, a significant amount of pre-systemic metabolism before tacrolimus even makes it to the portal vein to go to the liver. Now, we're all very aware within the liver, there's a very high concentration of a variety of cytochrome P450 enzymes in the liver. As you all are very aware, what is unusual in liver transplants is the cytochrome P450 system of the recipient in the gut is different from the cytochrome P450 system of the donor of which it was transplanted. And I'm not going to present you this data today, but I can share it with you that in the kidney transplant population, the recipient's genotype predicts the AUC, but in liver transplant population, the recipient genotype does not predict the overall exposure to tacrolimus. And so therefore, we all appreciate the continued elimination of tacrolimus via the bile duct and the variety of drug interactions that can occur when these drug transport proteins or these metabolic enzyme parameters are inhibited or induced. So taking all of this into account, we thought that it was very important for us to look at the effect here of the, I actually am sharing the data, the effect of the recipient CYP3A5 genotype does not impact the tacrolimus AUC in the liver transplant patients. Here you can see the expressors. So when you look at the recipient, okay, this is the recipient's genotype. Normally, you would expect to see these patients that are expressors have a lot lower tacrolimus exposure than the non-expressors. But you can see here that the recipient genotype does not impact the overall AUC exposure. In doing that, it was important for us to measure the 13-O-desmethyl component of tacrolimus, which is the highest concentration metabolite and also the metabolite with the most immunosuppressant activity. And you can see whether you're comparing AUC 0 to 12 or the AUC 0-12 ratio between the products. There's no difference between the products of the metabolite as well. And when we applied the stringent bioequivalence testing criteria, again, it passed bioequivalence testing standards. So in inclusion of this study, we observed that these formulations met both conventional average bioequivalence and reference scaled average bioequivalence standards in stable liver transplant patients with no difference in these pharmacokinetic parameters. There was comparable within subject variability for all the formulations. Although I didn't present it, there were no observed changes in organ function during the short 6 weeks period of this study, and there was a similar adverse event profile. And the various covariants that impact tacrolimus pharmacokinetics were no different by formulation. So now at this time, I'd like to unblind you to the results. So I refer to these products as product 1, product 2, and product 3. You can see here that product 1 was the brand product, product 2 was generic high, and product 3 was generic low. When you look at the product selection, the brand name was obviously Prograf. Generic high happened to be Adport, the Sandoz formulation, and generic low happened to be Dr. Reddy's formulation. And there were no statistically significant differences observed for any of the pharmacokinetic parameters by formulations in these liver transplant recipients. So in conclusion, I'll ask this provocative question. So what is the purpose of generic tacrolimus formulations if they're similar to brand? And it's obvious that there's a cost savings, but to whom is the cost savings afforded? It's primarily afforded to the provider or whomever is paying for the product, not necessarily to the transplant recipient who may be assuming any risk. If we decide as clinicians there is risk associated with using a generic product. And hopefully I've showed you that pharmacokinetically there shouldn't be. But again, as I stated, the experiment that you're starting in Egypt today, we started in the United States in 2009. I'm going to orient you to these two graphs for tacrolimus and for CellCept. And basically, this is time in years, January 10, 11, 12, 13, 14. And this is the percentage of formulation based upon these years that were used. The brand name is depicted in the solid line, and the generic is in the dashed line. However, what's important is these dashed vertical lines represent every time a new generic formulation was available on the marketplace. So you can see with the initial marketplace introduction of Adport or Sandoz tacrolimus in the United States, within, really less than a year, there was already a 50% conversion to generic product. And that percentage only continued to increase out to 2014. And that's as far as I have published data to show you. But as I told you, ironically, MMF became generic at the exact same time as tacrolimus in the United States. So while there were a variety of MMF formulations that were FDA approved, they were not allowed to be marketed for a variety of reasons until around July of 2009. And so then around July of 2009, you can see again, they're converted to a 50% amount of product uptake of the generic formulation in a shorter period of time than with tacrolimus. So for sake of time, I'm going to skip this and move on to the last slide, which talks about the cost of transplant of immunosuppressive agents after the introduction of generics. So in the United States, we're able to evaluate SRTR data in transplant recipients via our primary payer, which is Medicare Part D. And you can see the reduction in drug costs from 2008 to 2013 for tacrolimus for Medicare patients was roughly 50%, which translated to an annual cost savings of $1,700 to $2,700 per patient per year. For mycophenolate, there was a Medicare Part D payment decrease of 55 to 67%, with an average cost savings per patient of $2,500 to $4,200. And when we looked at patients that had unsubsidized coverage or were paying out of pocket for tacrolimus, there was a 61 to 72% cost reduction, resulting in $900 to $1,300 cost savings per patient. And with CellCept, a 71 to 79% decrease, resulting in $1,000 to $1,800 saving per patient. So annualized over a long period of time, it's a significant cost savings to the patient provider. So in conclusion, AdPort is bioequivalent to brand tacrolimus in healthy volunteers, which the data was not presented. AdPort is bioequivalent in stable liver transplant recipients with no observed changes in organ function or adverse event profile. Over 50% of the U.S. liver population had received generic tacrolimus and generic MMF since 2011, and the introduction of generic immunosuppressants have lowered the cost of immunosuppressive agents to the provider. And with that, I'll be happy to accept any questions.
Thank you very much, Professor Rita, for the very informative presentation. I think we bypassed a little bit the time, so I'll leave the floor for Professor Effat to introduce the second speaker, and then because in the interest of time, I think we're short of time.
Thank you very much, Dr. Rita Allowy. This was a very interesting presentation, and I think what we're discussing tonight is very important in our part of the world, with the cost of treatment and care here and around the world really rising to exponential levels. Let me now introduce Dr. Amit Kamar Rai, who is the Global Medical Manager of Sandoz Transplantation. His talk is about Sandoz mycophenolate mofetil, the clinical applications and outcomes in liver transplantation. Please go ahead, Dr. Rai.
Hello, good evening. My name is Amit Kumar Rai, and I'm the Global Medical Manager for anti-infectives, antivirals, as well as transplant franchise. And today I am here to discuss about mycophenolate mofetil, its executive summary and use in liver transplant. So before going into detail, let me just show you the agenda for today's presentation. So a small introduction about mycophenolate mofetil. Then next, I will discuss in brief about the importance of mycophenolate mofetil in liver transplant, followed by the clinical efficacy studies of mycophenolate mofetil and how the approval was granted. And at last, I will discuss the bioequivalent study of Sandoz mycophenolate mofetil in comparison to CellCept. Mycophenolate mofetil is the prodrug of mycophenolic acid and was developed to improve the bioavailability of the acid form. Commercially, mycophenolate is available in two different dosage forms. One is the mofetil form, which is the prodrug, and another is enteric-coated mycophenolate sodium. So after oral administration, the hydrolysis is taking place by esterases in gut wall, blood, liver, and tissues, and mycophenolic acid is formed, which is the active moiety. This is metabolized in liver, GI tract, and kidney by UGT enzyme to MPAG, which is mycophenolic acid glucuronide, and is the major metabolite of mycophenolate, which is excreted into urine via tubular secretion. But after a duration of almost 6 to 12 hours, there is an enterohepatic circulation taking place where MPAG is converted back to mycophenolic acid, and it almost covers 40% of the area under the curve for this. Coming to the mechanism of action and some PK/PD properties of this product. It's a potent, selective, and reversible non-competitive inhibitor of inosine monophosphate dehydrogenase, which is a key enzyme involved in the de novo synthesis of guanine nucleotides, hence leading to arrest of T and B lymphocyte proliferation. So in a nutshell, the proliferation of T and B lymphocytes is inhibited, and hence no antibody production happens, and hence the immunosuppressive action takes place. After oral administration, the absolute bioavailability ranges from 81% to 94%. MPA, the acid form, is almost 99% bound to serum albumin in patients with normal liver and renal functions. It displays a linear pharmacokinetic with Cmax occurring in 1 to 2 hours. And as discussed previously, around 6 to 12 hours, a secondary peak is observed due to enterohepatic circulation, which is almost 40% of the AUC. The mean elimination half-life of this product ranges from 9 to 17 hours. Coming to approval indications, dosing, and most common adverse events. MMF was approved in the United States in 1995 for prophylaxis of organ rejection in kidney, heart, and liver transplant recipients. The similar approval was granted in 1996 in EU. The approval was based initially on the data from 3 randomized double-blind controlled phase 3 trials in de novo kidney transplant patients. And later there was a trial which I will discuss today for the liver transplant patients also. It is marketed under the brand name Cellcept by Roche Pharmaceuticals. Coming to the dosing section, in adult renal transplant recipient, 1 gram twice daily. In adult hepatic transplant, it's 1 gram twice daily IV or 1.5 gram twice daily or orally. In cardiac transplant recipients, it's 1.5 gram twice daily orally or IV. To get more detailed information on the dosing regimen, I would urge to have a look on this summary of product characteristics for either Cellcept or for Sandoz mycophenolate mofetil. Coming to the adverse events part, most relevant adverse events occur in GI tract, which are nausea, vomiting, diarrhea, abdominal pain, and gastritis, which might occur in up to 45% of the patients. And are often a reason for dose reduction or discontinuation. But these events can be easily managed just by reducing the dose. So coming to the importance of MMF in liver transplant, after the first approval in 1995, it became the first line drug for the treatment for the transplantation. An estimated 79% kidney, 48% liver, and 75% of the heart transplant recipients are now prescribed mycophenolate instead of azathioprine at hospital discharge. It is now widely used immunosuppressant, especially in calcineurin inhibitor sparing regimen, because we know that CNI inhibitors are toxic, they are nephrotoxic, can cause diabetes mellitus and also de novo malignancies in long-term use. Hence, now MMF is widely used to circumvent the CNI inhibitors side effects. It improves both graft and patient survival in liver transplant patients, which we will see in the efficacy studies. Almost 77% of all liver transplant patients in the United States receive MMF at the time of discharge from hospital and is most commonly used with tacrolimus and steroids. Here in the table from 1999 to 2008, you can see that the percentage of patients discharged at tacrolimus, MMF, and corticosteroids almost doubled in 2008. So moving on now to the efficacy studies of MMF in liver transplant patients. Here I will discuss 3 different studies. This is the first study in which MMF is compared with azathioprine in a double-blind randomized trial. And this is the pivotal trial which led to the approval of MMF in liver transplantation. This study involved 565 primary liver transplant patients, and they were randomized to MMF group, 278 patients in MMF group, and 287 patients in azathioprine group. And they were followed up till 1 year. Coming to the results, the incidence of acute rejection or graft loss was less, which is 38.5% in MMF patients compared to almost 48% in azathioprine patients. The incidence of biopsy-proven acute rejection was less in MMF group, 31% compared to 40% in azathioprine group. But 1 year post-transplantation, the patient and graft survival rates in both MMF as well as other azathioprine group was similar. Here also you can see that the percentage of incidence for time to rejection censoring for graft loss is better in MMF group compared to azathioprine group. Hence, in conclusion, MMF was superior to azathioprine, preventing acute rejection in the first 6 months post-transplantation. But after 1 year, it was equivalent in preventing graft loss, similar to azathioprine group. Now, coming to the second study, which is a retrospective study of 11,600 patients, of which 3,463 patients were hepatitis C positive. One of the major reasons for the liver transplant is hepatitis C. Hence, this is an important study to discuss here. Patients who were discharged from hospital with tacrolimus-based immunosuppressants with MMF, so patients were randomized in 2 different groups, one with MMF and one without MMF, just to see its effect. So coming to the results section, recipients treated at discharge with MMF, tacrolimus, and corticosteroids had increased patient survival, 81% versus 77% without MMF, increased graft survival, 76% versus 72% comparison without MMF group, as well as lower rates of acute rejection. In multiple regression analysis, MMF triple therapy at discharge was also associated with a reduced risk of death, graft loss, acute rejection, death from infectious complication when compared to the group which was not prescribed MMF. So here also you can see in these graphs that the patient survival overall was better in case of the triple therapy, which involved MMF, compared to therapy where no MMF was prescribed. Here, a similar sort of graph is for patients with hepatitis C, where the patient survival was better in group having treated with MMF when compared to without MMF. So overall, addition of MMF at discharge to tacrolimus-based immunosuppression is associated with long-term outcomes after liver transplantation in patients both with and without HCV. Moving on to the last efficacy. Yeah. Sorry. Yes. Okay. So moving on to the last efficacy study. This is just to show that MMF is also better in lowering the dose of CNI, so that the side effects can be minimized. And here, this study just compared reduced dose tacrolimus with MMF versus standard dose. And in result, it was found that acute graft rejection as well as occurred less in group in which there was a reduction in tacrolimus dose with MMF compared to no reduction. And also the renal dysfunction, arterial hypertension or diabetes occurred less in group where tacrolimus dose was reduced along with the MMF. So this is just to show the effect of low tacrolimus dose along with the MMF. It improves the occurrence of acute graft rejection. In conclusion, reduced dose tacrolimus with MMF reduces the occurrence of renal dysfunction and the risk of graft rejection, and hence has the potential to replace full dose tacrolimus in adult liver transplantation. Coming to the BE study of Sandoz MMF versus Celcept. Sorry. Yeah, so here in this bioequivalence study, Sandoz mycophenolate mofetil was compared with Celcept. In total, 57 healthy patients were included in this study. And as you can see, the plasma concentration time graph for mycophenolate mofetil from Sandoz as well as Celcept overlap properly. And the 90% ratio of the geometric mean also falls within the 90 to 125 prescribed by the health authorities. Hence, based on this, the approval was granted for Sandoz mycophenolate mofetil. No serious adverse events was also reported during this study, and both the formulations were well tolerated. And hence, to conclude, based on these results, Sandoz mycophenolate mofetil was bioequivalent to Celcept. And at last, I just want to show that Sandoz MMF is also FDA approved. And with this last slide of mine, I just want to show the importance of the also support the use of generic in transplant patients, because generics are approved with strict standards and are bioequivalent to originator product. With this, I would like to thank you for all the time and attention. Thank you.
Thank you very much, Dr. Rai, for your nice presentation. I think now the floor is open for discussion. Okay. I'd like to thank Dr. Rai for his very nice presentation, and I'd like to thank Dr. Rita Allowy for her excellent presentation. It was really enjoyable. And actually, I cannot stress how much the economics of therapeutics are just as important as the efficiency. But the efficiency comes first. And I think both the speakers today have demonstrated and stressed the efficiency of these medications, especially when you're dealing with the very precious liver transplant patients. And I must admit that we always hesitate in modifying any treatments when it comes to these precious patients, and especially when they receive special gifts from related donors that give them part of their livers. So actually, you've set the floor for a very nice way of thinking that should move ahead, especially when we talk about the finances of this country and the finances of treatment, which are very heavy in general. We have few questions from the panel, and the panel has been the audience has been very patient today, and there are a few questions from the audience actually. One of them is, how do you manage, I think this is to Dr. Rita, how do you manage interpatient and intrapatient variability? Does it differ in the generic than the brands? And a related question to it is, why do you do your studies, which I think the answer is obvious, of course, on healthy individuals and not on recipients to start with? Dr. Rita, please go ahead.
Okay, so I'll take the second question first. It is important to establish the healthy volunteer data to understand if a product is bioequivalent or not. Obviously, before you introduce it to a transplant recipient. While it is vilified, the process of healthy volunteer testing with just a single dose is vilified sometimes, as I said. It's actually very precise when you look at the calculations of which they conduct on those limited number of patients. And if you do more patients and more healthy volunteers, it allows you to have a less equivalent product to actually meet the standards in that way. So I think it's always important to first start in healthy volunteers anytime for any therapeutic that you're using before you do it in patients, especially transplant patients. The second question, in terms of intra and interpatient variability, I think the data speaks for itself in regard to interpatient variability, okay? Within subject variability, so intra-patient variability. It is really quite small when it's very controlled, all right? When we see variability in a clinical setting and we think that it is associated to product, you have to understand there are many sources of variability. You know, what the patient had for dinner the night before, was it a high-fat meal or was it a salad? You know, what time did they actually take it? When did they get their blood drawn? Had they been compliant for the previous days? So there are actually a lot of factors that impact the intra-patient variability when we deal with it from a clinical perspective. In terms of interpatient variability, I think we all really know the main factor that drives that is the gene expression of the CYP3A4 in not only the liver and the gut. The patients that are high expressors are going to require anywhere from 2 to 3 times the dose of a low or a non-expressor. And that's where we see primarily the differences in inter-patient variability that is observed.
Thank you very much. I don't see Dr. El Meteini is here with us, or he?
Yes.
Yes. Do you have anything to?
No, thank you, Effat. I just want to thank both speakers for their very nice talks and very informative about both drugs. And if I may ask Rita about the study have been mainly after or in liver transplant patients who have stabilized after a certain period of time. In fresh liver transplants, do you have any figure about how many fresh liver transplants would use the generic tacrolimus from day 0, from day 1?
In the United States, you never say 100%, but in the United States, I would argue that it would be definitely above 80%, because all of that is administered in the hospital setting, of which hospitals are sometimes on a lot more restricted formulary than maybe the patient will be able to get brand based upon their insurance when they leave. But in the United States, where cost is an issue and hospital pharmacy is driving down the cost, most of them that I know of are using the generic products. There may be a few isolated cases which still request brand, but at this point in time, the debate has kind of settled. You know, in the United States, the debate was really, really hot from 2009 to maybe 2013 or 14. Now it's not really up for discussion. It's hardly discussed at all anymore because the use of generics is so frequent.
Yeah. Agreed.
You know,
I think. Yeah, please go ahead.
No, it's okay. Go ahead.
Go ahead, Dr. Mahmoud, please.
For Ahmed, the question also for the MMF and the formulation, the Sandoz MMF, is it in 250 and 500 available dose?
Yeah.
Same.
Yeah.
250 and 500. Okay. And again, the percentage in the liver world that is using the generic Sandoz as we speak now. Do you have any figures to compare?
Actually, I do not have any figures. I would agree to what just Professor Rita mentioned. But here in Europe, the payer system is totally different from what we have in the United States. So every European country has its own way of defining the price reduction in generic. So I think this also plays a major role. I do not recall the name of the country, but I think it's France or Belgium, where the price difference between the brand and the generics are very low, are not so much different. But I think if it is a hospital setting, generics are always preferred when compared to the brand. Thank you.
May I ask?
Yeah, go ahead.
May I ask you what's the philosophy for no difference in price in between the generics and the original?
I actually this is actually a good question. It's I really don't know, because I don't know. It depends on the healthcare policies of each country, because in some countries it's tender based. In some country is I don't know. I have no proper answer for this.
I can see Rita can answer this question, but just if there is no price difference, wouldn't this negate the benefit of any generic?
But this case is only applicable for transplant. But in other therapeutic areas, there is a huge price reduction for generic.
I speak about the transplant, the liver transplant.
Yeah. Yes,
well, Rita, maybe
I mean they are absolutely not equal, but the reduction is not so much when I compare it with other therapeutic areas. So I don't know the reason.
Yeah, but is this scenario in the US as well, or is there a difference in price in the US?
Huge difference.
Huge. Yeah.
So typically when generics are introduced to the market, you see a 75% reduction in price compared to the brand. I think what we've noticed with tacrolimus and CellCept, we've really only observed approximately a 50% cost reduction, but still it is a significant savings. In regards to the initial question related to France, I obviously I'm not an expert with France, but I think that pharmaceutical companies, when their products go off patent and go on to generic, they have two different philosophies. Some say basically, we're going to reduce our price and compete with generics. We can make them just as cheaply as they can, and we'll keep selling and maintain our market share. You have another company philosophy that is we believe in our brand. We believe that there's brand name support, and we are going to stick with brand. To some extent, the prices continue to increase with the brand, even after a generic has been introduced. So I think it's a combination of reasons as been discussed. It's the brand name manufacturer's philosophy toward generic market penetration, and then also the specific country's payer system.
Thank you very much.
And their political forces, obviously, as well.
Absolutely.
Of course. I believe in Egypt, from what I understand, it is about 50% less in tacrolimus, and it's about 30% for MMF, which is a big amount of money to be saved in a country like ours, where we have a big volume of liver and kidney transplants, and we hope to do more in the future, and where the budgets are already stretched. So there is a big place for generics in Egypt and many other countries. I really thank you for this very nice subject and out of the box, really, subject that is, and I congratulate Sandoz for penetrating this domain and opening new era for transplantation in Egypt, where cost matters as much as results. Dr. Mahmoud, do you have anything to add?
No, I think we bypassed our time. Now we are approaching 2 hours. So thank you very much, both speakers, Dr. Rita and Dr. Amit, my dear friend, Professor Effat. And I would like to thank the Sandoz company and Sandoz team for preparing this webinar. I don't know how many followers or how many participants do we have who are saying at least we are 9 till now. It's getting late and it's time for dinner. So thank you very much, and I hope that we'll meet again very soon. And as Rifat just alluded to, I think these new products, the two new products will make us a lot of saving in the transplant business that we needed at this difficult time and the challenging time in the COVID and the post-COVID area. And maybe another topic that we would like to meet up shortly about because we have suspended our LDLT program, both of us, over the past few weeks. So how are we going to reopen now and very shortly, and what are the challenges and what are the precautions and what we should so cost effectiveness is one important aspect of it. So you hit at this point, and I hope we'll meet shortly with another topic. Thank you all.
Thank you very much.
Thank you.